Assalamualaikum and good morning, and welcome to this morning's episode of Breakfast at UMHealth. Without further ado, let me introduce the first speaker, Dr. Aslina Pahrudin Arrozi. Dr. Aslina is a newly appointed senior lecturer at the Department of Molecular Medicine. She did her undergraduate degree in biochemistry and master's in medical science both at University Kebangsaan Malaysia, and obtained her PhD in medical science from Shiga University of Medical Science in Japan. Her current research interest focuses on the pathogenesis of Alzheimer's disease and the neuroprotective strategies through natural products. So the title of our talk today is Deciphering Alzheimer's Complexity and Neuroprotective Strategies Through Natural Products. The floor is yours, Aslina.
Thank you, Prof. Aslina, for the introduction. Assalamualaikum and good morning, everyone. So my presentation today will consist of two parts. The first part will cover the fundamental components of Alzheimer's disease, including its etiology, pathogenesis, diagnosis, and current treatment. And the second part will focus on the beneficial health properties of utilizing natural products as potential treatments for neurodegenerative conditions. This is an overview of Alzheimer's disease. So what happened in Alzheimer's disease is that the neuron cells are damaged. And one of the first areas in the brain affected is the hippocampus. The hippocampus in the brain is responsible for memory, language, and thinking. As a result, the first symptoms in Alzheimer's disease tend to be memory, language, and thinking problems. Although these symptoms are new to the individual affected, the brain changes that cause them are thought to begin 20 years or more before the symptoms develop. Now, I would like to share the prevalence of Alzheimer's disease. The World Alzheimer's Report 2021 revealed that over 50 million people globally live with dementia, the majority of which 60 to 70% corresponds to Alzheimer's disease. In addition, the majority of the cases were reported in low and middle income countries. In Malaysia, data on the prevalence of dementia at the national level has not been updated. However, according to the Alzheimer's Disease International 2015, 123,000 cases, 123,000 individuals were living with Alzheimer's, and this figure was expected to double by 2030. In 2015, 60% of the cases suffers the end stage, where the individual will wander around and needs to be located within 24 hours. Unfortunately, half of the 60% will face serious injury or death. In terms of gender, women constitute a larger share of Alzheimer's patients than men and have greater risk of developing the disease as they age. In 2019, prior to the onset of COVID-19, Alzheimer's disease and other dementias was ranked as the 7th leading cause of death worldwide and among females in Malaysia. And a population-based survey conducted by the Institute of Public Health in Malaysia reported that the prevalence of dementia among older persons aged 60 years and over was estimated to be 8.5%. These findings emphasize the significant burden of dementia care in Malaysia, as WHO estimates the prevalence of dementia to be between 5% and 8% among the general older adult population. When talking about Alzheimer's, many people sometimes confuse between Alzheimer's and dementia. Dementia is not a disease, but it is an umbrella term for anything that can cause an issue with brain functioning, including confusion, memory loss, or the loss of problem solving. And Alzheimer's is a disease, and it is the most common cause of dementia, accounted 60 to 70% cases worldwide. There are other causes of dementia, such as vascular dementia, dementia with Lewy bodies, Parkinson's dementia, and frontotemporal dementia. And it is important to note that some individuals may show dementia-like symptoms. However, it is not considered as dementia without progressive brain changes. And the causes of dementia-like symptoms include depressions and the effects of medications, and these conditions often may be reversed with treatment. One of the challenges when addressing Alzheimer's in the general public is there is always misconception with normal aging. While memory loss could happen in both Alzheimer's and normal aging, there are big differences between them. For example, in normal aging, only certain aspects of memories are forgotten, and these memories can often be recalled and improved with cues and context. In contrast, in Alzheimer's disease, the challenge extends beyond forgetting entire experiences. Even recent memories are difficult to retrieve despite assistance with cues and context. So it is important to highlight here that Alzheimer's disease is not caused by aging and is not a normal part of the aging process. Instead, aging is the main risk factors in the late onset of Alzheimer's disease. There are two main classifications of Alzheimer's: the late onset, which often diagnosed after the age of 60, and the early onset or familial, which typically identified before the age of 60. The sporadic Alzheimer's is the most common form, and it does not have a clear genetic cause and may occur randomly in older individuals. Combination of genetic, environments, and lifestyle may contribute to the cause of sporadic Alzheimer. In contrast, the familial Alzheimer's is caused by the genetic mutations in genes that control the production of pathological protein known as amyloid beta. The familial AD, however, is only represent less than 10% of all AD cases. So in general, Alzheimer's is a complex disorder due to the fact that most of the cases are sporadic, which is arise from multiple factors rather than a single cause. I will talk more about these amyloid beta proteins later in my slide, but for now, remember that this amyloid beta protein is the main component of amyloid plaques found in the brain of patients with Alzheimer's disease. The risk factor for sporadic Alzheimer's can be grouped as non-modifiable and modifiable. The greatest risk factors, as mentioned earlier, is an older age. Genetic and having a family history with Alzheimer's are also the non-modifiable risk factors. And the risk factors involving the genetic is mostly related to genetic factors that contribute to the APP metabolisms and amyloid beta regulations. The modifiable risk factors are factors that we can change to reduce the risk. Examples are physical activity, education, health factors, and cognitive engagement. Alzheimer's disease is a progressive disease, meaning it gets worse with time. And the progression of Alzheimer's disease is usually from brain changes that are unnoticeable by the person affected to brain changes that cause memory problems and eventually physical disabilities. This progression is what we call AD continuum. On this continuum, there are three broad phases: the preclinical AD, which showed no symptoms but possible biological changes in the brain. And second is preclinical Alzheimer's disease. The second is mild cognitive impairments, and the third is Alzheimer's disease, which can be further broken down into mild, moderate, and severe. While individuals with mild cognitive impairments may often continue to work, drive, and participate in their favorite activities with occasional help from family members and friends, no one knows how quickly the disease may progress to the next stage. As time passes, more neurons are damaged and more areas of the brain are affected. Ultimately, Alzheimer's disease is fatal, and studies indicate that the average life expectancy for a person with Alzheimer's disease is 5.8 years after diagnosis, yet some live as long as 20 years. And the length of each part of the continuum is vary from person to person, and it's often influenced by many factors. Until now, Alzheimer's disease remains an unsolved mystery. However, the field of Alzheimer's research is far from stagnant. The human brain is an incredibly intricate organ, and unraveling the precise mechanism of disease that affects its structure and functions is no small feat. The evidence of the pathology in Alzheimer's disease is all there. The neuron showed accumulations of amyloid plaque extracellularly, tau tangles intracellularly, rampant oxidative damage, synaptic dysfunction, neuroinflammations, damaged mitochondria, defective calcium homeostasis, and etc. Although the prime suspect, which is the accumulation of beta amyloid is easy to identify. Until now, no one has been able to pin the suspect to the crime. So when looking at the structure of the normal neurons, the basic part will consist of cell body, multiple dendrites, and axon. And this extension enable individual neurons to form connections with other neurons. At such connection called synapse, information flows in tiny bursts of chemicals that are released by one neuron and taken up by another neuron. And this allow the signals to travel rapidly through the brain. And these signals create the cellular basis for memories, thought, sensations, emotions, and movements, as well as skills. However, in Alzheimer's disease, plaques and smaller accumulations of beta amyloid outside neurons interrupt with the neuron-to-neuron communication at the synapse, whereas inside neurons, tau tangles block the transportation of nutrients and other molecules essential for the normal function and survival of neurons. Together, these proteins may induce neurodegeneration or neuronal death. This amyloid tau neurodegeneration framework makes up the key pathology in Alzheimer's disease. So this slide actually want to show how does the amyloid beta plaque is formed, but it is only in a simple way. So actually, amyloid beta is derived from amyloid precursor protein, which is found in the membrane of neurons and plays an important role in neuron growth and repair after an injury. In physiological condition without a disease, the APP protein is usually processed via non-amylogenic pathway without plaque formations. And this pathway usually leads to a release of products which has a neuroprotective properties. However, in Alzheimer's disease, the amyloidogenic pathway takes over, where the APP is cleaved by beta secretase and gamma secretase, resulting the formation of the fragments of amyloid beta, which are prone to aggregation and form amyloid plaques in the extracellular space. So this is why in familial Alzheimer's disease, genetic mutations in genes such as APP and presenilin, which are the main subunit in gamma secretase, will favor the amyloidogenic pathway in producing the toxic amyloid beta. And this amyloid beta has multiple variants, but the amyloid beta 42 is considered as the most toxic form. Next is about tau tangles. Tau is a protein contained within the axons of the neurons, and this protein is encoded by the MECT gene. Tau stabilizes the microtubules and essential structures that transport nutrients within neuron. In a healthy brain, the proteins help these microtubules remain straight and strong. But in Alzheimer's disease, tau dissociate from the microtubules and the microtubule destabilize. One of the causes of the dissociation from microtubules is hyperphosphorylation, and the dissociated tau will bind together to form oligomer and to form helical filaments and eventually to form neurofibrillary tangles. The genetic mutations in MECT gene, however, are not linked to familial Alzheimer's, but it can cause other dementia collectively called tauopathies. And Alzheimer's disease also considered as secondary tauopathy, as both tau and amyloid beta are present. In Alzheimer's disease diagnosis, amyloid beta and tau are the core and specific biomarkers, and this figure showed the immunohistostaining of postmortem brain tissue samples. The AD brain exhibits positive staining for amyloid beta and tau antibodies, and it is observable that amyloid beta is deposited outside the neuron cells, while tau tangles are found inside the neuron cells. Over the past 10 years, both amyloid beta and tau have been demonstrated to self-propagate and spread throughout the brain by prion-like mechanisms. And interesting differences between amyloid beta and tau is that amyloid beta is detected before tau and even before the clinical onset. It is even reported that accumulation of amyloid beta may result in subsequent increase of tau. However, tau is more correlated with the clinical onset of the disease than amyloid beta. The Bragg staging to classify the degree of pathology in Alzheimer's disease use phospho-tau. Therefore, tau has been widely used as prognostic markers of future cognitive decline in cognitively normal to demented individuals. As the understanding of Alzheimer's disease pathology has developed, the clinical diagnostic standard has underwent revision with the latest occurring in 2018. The detection of amyloid beta and tau biomarkers in living patients traditionally involves cerebrospinal fluid, CSF, and subsequently extend to peripheral blood and saliva. Additionally, there are emerging biomarkers for Alzheimer's disease found in other body fluids, including tears and nasal. A recent study, it is indicated that older adults with olfactory dysfunction were more than twice as likely to develop dementia 5 years later, and more order identification error were associated with a greater probability of an interval dementia diagnosis. However, due to low amount of amyloid beta and tau in nasal, a highly sensitive method to discriminate between AD and non-AD is required. This is what I did during my previous study, where my colleagues and I developed an alternative method for detecting tau in nasal samples and compare it with ELISA. This method is incorporate a tau cell biosensor that is able to amplify abnormal tau even when present in low amounts in the samples. The concept behind this approach is to take the advantage of the properties of abnormal tau, which can self-propagate and induce naive tau to misfold and thereby producing new tau seeds. While we demonstrated that this method is more effective in discriminating AD and non-AD compared to ELISA, it still requires improvements to enhance reliability and feasibility. Until now, me and my colleagues in Japan are still working on this project. Now, I would like to talk about the AD treatments. Several prescription drugs are approved by the US FDA to help either manage the symptoms or to treat the disease. And most of FDA approved drugs work best for people in the early or middle stage of Alzheimer's. There are currently no known interventions that will cure Alzheimer's. And the FDA has given accelerated approval for aducanumab in 2021, which is the first disease-modifying therapy for Alzheimer's disease. The medication helped to reduce amyloid deposits in the brain. Lecanemab is the second FDA-approved drug in 2023, and donanemab is still pending review from FDA. All these three monoclonal antibody-based drugs are targeting amyloid beta. However, they work differently by targeting amyloid beta at different stages of plaque formations. The ultimate objective is to lower the beta amyloid so that to slow the progression of the disease and to reduce the clinical decline. According to Arrozi Forum in March 2023, 298 AD therapies undergo clinical trials, and 76 of them target the amyloid beta peptide or its aggregate. So far, only 2 antibody-based drugs, aducanumab and lecanemab, have been approved by the FDA, and 38 of them have been discontinued due to ineffectiveness or toxic side effects. Most clinical drug trials target at amyloid beta and tau, but the clinical failures of the recent decades indicate that there are further pathological mechanisms. So, in effort to discover new strategies for AD therapy, research community and the pharmaceutical industry are moving towards the development of multi-targeted drugs, which can target different pathological mechanisms associated with Alzheimer's disease. And one of the preferable strategies in Alzheimer's disease drug discovery is using natural products. Multiple bioactive compounds in natural products has the ability to exert multiple neuroprotective mechanisms, and the observed neuroprotective effects have typically been recognized as being antioxidative, anti-neuroinflammatory, anti-amyloid beta aggregation, anti-tau proteins, and enhancing cholinergic signaling. It is reasonable to speculate that the onset and progression of Alzheimer's disease could be slowed down or even prevented by natural products working on multiple pathological targets. These are the list of some of the natural products for Alzheimer's disease, and there are natural products that has entered clinical trials. For example, root extracts of Panax ginseng. A study reported that ginseng extract administered long-term gradually improved cognitive functions with minor side effects on AD patients. And another study demonstrated that the efficacy of ginseng extract on cognitive functions was related to the increase of relative EEG alpha power. Besides ginseng, studies have revealed a possible link between regular intake of turmeric as part of the curry and excellent cognitive performance among the elderly. In AD patients, turmeric treatment greatly improved their behavioral symptoms, supporting the probable benefits of the use of turmeric in individuals with Alzheimer's disease. Furthermore, it has been demonstrated that natural products found in nuts such as walnuts, almonds, and hazelnuts have beneficial effects on neuroprotections against Alzheimer's disease. And several in vivo studies have confirmed the relationship between nut consumption, improved cognitive performance, and reduced incidence of Alzheimer's disease, thereby supporting the preventive properties of nuts against Alzheimer's disease. Several marine natural products have also demonstrated effective outcomes against Alzheimer's disease. For example, decoy extracted from brown algae has exhibited potent inhibitory effects on amyloid beta accumulation, both within and outside of cells. Diterpinone found in marine sponge can inhibit the BACE1 enzyme in the amyloidogenic pathway and reduce the hypophosphorylation of tau protein. Additionally, homotaurin, derived from red seaweeds and possessing a structure similar to the neurotransmitter GABA, has been shown to safely decrease the concentrations of amyloid beta 42 in CSF in phase 2 clinical trials involving MCI patients, offering potential for disease improvement. Next is the list of the natural substances that are being included in the upcoming clinical trials for Alzheimer's disease drug development in the USA. Huperzine from Huperzia and sodium oligomannate from brown algae are currently undergoing phase 4 trials, while ginkgo ginkgolide and bilobolide from ginkgo biloba are in phase 3 trials. Sodium oligomannate is among the first to address the gut-brain axis, a recent focus of research in neurodegenerative diseases. The available data currently support its influence on the gut microbiome, leading to an improvement in AD-related symptoms. The grape seed extract from grape is in phase 2 trials, and elderberry juice from elderberries has recently finished its trials. PDPP, a blend of grapeseed extract and resveratrol, has recently entered phase 1 trials. Together, the distinct properties of these compounds derived from natural sources should continue to promote the assessment of combination therapies such as anti-amyloid beta immunotherapy and treatment with natural products. While natural products and their isolated natural compounds have been well studied as neuroprotective agents and valuable resources for exploring novel approach in AD treatments, many of them still remain untested, and their clinical use is not easy to monitor for a wide variety of reasons, such as the quality of the natural source, bioavailability, capacity to reach the central nervous system, and the ability to cross the blood-brain barrier. However, new approaches and strategies to help promote the CNA access of this neuroprotective agent, such as the incorporation of nanotechnology in the delivery of natural products, has helped to ensure the safety and efficacy of natural products therapies. Example here, the nanolipidic green tea polyphenol EGCG particles increase its oral bioavailability in vivo more than twofold over free EGCG, along with increase in alpha secretase activity, that is the enzyme responsible to drive the non-amyloidogenic pathway. I also would like to share some of the projects that I did during my master's and PhD studies. I have actively participated in various projects centered around investigating neuroprotective strategies for Alzheimer's disease. The vitamin E in Alzheimer's disease are widely recognized However, limited knowledge exists regarding the efficacy of the vitamin E individual isomers. So what I did in my master study is a comparative analysis between vitamin E isomers derived from Malaysian palm oil, and the result revealed that gamma tocopherol exhibit more pronounced effects than alpha tocopherol in modulating the amyloidogenic pathway and mitochondrial functions in vitro. Furthermore, we also developed derivatives of curcumin referred to Shigawai within our own laboratory in Japan. Our experiments reveal inhibitory effects on the TXNIP protein within the brains of animal models with Alzheimer's disease, indicating its potential to regulate cellular stress induced by TXNIP. Additionally, we also have partnered with Morinaga, a Japanese F&B company specializing in meat-based products, and our collaboration has revealed that MKP found in meat peptide demonstrate neuroprotective effects by enhancing cognitive functions and reduce inflammation in the brains of animal models with Alzheimer's disease. As of now, there are still 2 projects in progress with my role as a co-researcher, and this project was initiated during my tenure as an assistant professor at Shiga University of Medical Science in 2022. And the first project is about the TRF on the gut microbiome and association with cognitive functions. This project is just a preliminary study, and we already completed obtaining all the samples. So right now is at the stage of sample processing and data analysis. And the second project is the use of red ginseng extracts in FPP-PS1 mice models. This project is already finished at in vivo at the preclinical levels, but the data is not yet published as of now. And our colleagues in Japan are now want to further this red ginseng extract to enter the phase 1 and 2 clinical trials. Currently, I'm engaged in active collaboration with researchers from Shiga University of Medical Science in Japan and UKM, and I look forward to expanding my collaborations with additional partners in the future. I'm open to collaborating on research with any researchers exploring intriguing avenues in these fields. So I have finished my talk with that. Thank you for listening.
Thank you very much, Aslina, for the insightful talk on Alzheimer's disease. There's actually one question in the chat box. Are we able to send for AD blood or CSF biomarkers at UMMC? You probably won't have the answer to this, but perhaps you can share a little bit how would you measure you talk about the 2 biomarkers, right? The amyloid beta and tau. So what's the methods that you use to measure these biomarkers, Aslina?
The conventional method is by ELISA. Yeah, but sometimes the result is not. They give how to say, like many different kind of result because of the limitation of the method. So that's why ELISA could give different result in different setting of the lab. So right now we are actively searching for the more sensitive method to measure the Alzheimer's disease biomarker in the blood or any other body fluids that are less invasive. Yeah.
I hope that answers your question for the first person. There's another question. As a senior, what supplements would you recommend? There are so many. Do you think supplements would help, Aslina?
It depends actually, because Alzheimer's is a progressive disease, so it depends at what stage you are. So I don't think I can recommend the best supplements, but so far the best thing is to prevent rather than to treat. So I believe that any supplements using natural products, that if you are taking it at the earliest stage before you are getting older, so maybe it can prevent you from getting the disease. And of course, like I mentioned previously, Alzheimer's disease have many factors, and one of the interesting factors is actually the education levels. There are studies that have already showed that high educations may lower the risk of Alzheimer's disease. Yeah, something like this. So maybe you have to use your brain more in order to prevent you from getting the disease.
Because you mentioned about the studies, people have shown that consuming nuts can actually be good,
Yes.
right?
Yeah.
So do you know if there's any certain amounts of nuts that you have to consume? Is it okay that if we just consume it as nut as it is, or it has to be in some kind of concentrated form?
So far, there is no study to mention which kind of nuts that is will give a better effect, but I think as long as we take it as in a considerate amount without, like everything is must be in a considerate amount. So it cannot be more than the usual, or it cannot be overdose, I think.
Okay, there's another question regarding follow-up question on that supplements, yeah, Aslina. So the questioner was asking that many people take lion mane, mushroom, ginkgo, coconut oil as a supplement. Do you know if any of these can actually be useful for preventive effects?
Coconut oil, there is some study that already started to test it in for the Alzheimer's disease, but the rest is maybe I not really sure.
Okay, Aslina, since we're already 1 minute past the time, there is actually another question. Perhaps you could answer that offline by typing in the chat box.
Okay.
Okay, so with that, I thank you, Aslina, for sharing on the research that you are doing currently, and wishing you all the best. I now pass over to the next moderator. Thank you very much.